Good day. So in this session, I am to talk about the basic concepts of immunology and serology. So first, let us define the terms. Immunology means it's the study of a host reactions when foreign substances are introduced into the body. So commonly, these foreign substances are what we call as the antigens, because immunology is a study of how the molecules, the cells, the organs, and the systems in our body recognize and dispose this foreign material. So that means it's the study of the recognition and disposal of these foreign substances, what we call as the antigens. So in short, it deals with the body components, how it responds, how it interacts, and the undesirable consequences of these immune interactions. And also, it could be a study of how our immune system can be manipulated to protect us against certain diseases. And also, this is an area of biology concerned with the processes by which all living organisms defend themselves against infection, because our immune system can recognize which is belonging to ourself or which is a non-self antigen. So by knowing the non-self antigens, it could defend us or it could defend our body against non-self materials or non-self antigens. Next one, it comes from the Latin word immunis, that means exempt, or immunitas, meaning to say freedom from. So that means immunology is concerned with how our immune system would protect us from certain diseases. How about this one? Serology. So it comes from the Latin word sero, or meaning to say serum. So it's the branch of science dealing with the measurement and characterization of antibodies, antigens, and other immunological substances in the body fluids, particularly in our serum. So in short, it deals with our serum to know their immunological reactions and properties. So when we say immunology and serology, so that means this is the study of how the immune system are organized and how they function, and of course the different serological techniques. So for the general roles of the immune system, we have first defending the body against infection, and also in the recognition of foreign antigens and how the immune system would respond to these foreign antigens whenever they are introduced to the body, and of course the disposal of these foreign antigens. And lastly, defending the body against the development of tumors. So although our immune system is composed of a large, complex set of widely distributed elements, it is still distinctive in its characteristic, just like specificity and memory. So particularly, this includes the production of antibodies whenever we are exposed to foreign antigens. And this specificity and memory are characteristics of lymphocytes. And also, the various specific and non-specific elements of the immune system can demonstrate mobility. That is the reason why sometimes if we have local sensitization, just like an insect bite, it could result to systemic sensitization, because again, the cells of the immune system are having this mobility. So that includes the T and B lymphocytes. Also, it includes the antibodies or the immunoglobulins, the complement, and other hematopoietic cells. Also, the cellular components of the immune system can replicate. So replicability. And lastly, there is a cooperation between cells or cellular products. So this cooperative interaction involves specific cellular elements and other non-lymphoid elements, and also cellular products. Let's have the brief history of immunology. So the first written records date back to 1500s, when the Chinese developed a practice of inhaling powder from smallpox lesions or scabs. So smallpox lesions or scabs in order to produce protection from this dreaded disease. And this one, the smallpox, it is caused by a virus known as variola. That's why the practice of deliberately exposing an individual to material from smallpox lesion was known as variolation. So variolation. And the theory was that if an individual was exposed in a healthy state as a child or young adult, the effects of the disease would be minimized. So it's as if the immune system was oriented to recognize the virus coming from the smallpox lesion. So if a person was inflicted with a disease, the consequences of the disease will not be that serious. And also, in 1718, the practice of variolation was introduced to Eastern and Western medicine by Lady Montagu. So Lady Montagu came from a wealthy and entitled family, and she was actually a daughter of a duke. And smallpox killed her favorite brother. Then 2 years later, it also inflicted her. So Lady Montagu was very brilliant and attractive woman, but this disease left her with deep and pitted scars. So maybe that's what inspired her to educate people, particularly in England and some Eastern and Western countries, because during this time, this smallpox disease was the deadliest, wiping out more people than the Black Plague. And another thing, the last case recorded of this smallpox disease was on October 1977. And it was only in 1980 when the World Health Organization certified the eradication of smallpox. So next one, in the late 1700s, an English country doctor named as Edward Jenner discovered a remarkable relationship between exposure to cowpox and immunity to smallpox. So he showed that when you are exposed to a cowpox virus, you will be protected from a smallpox disease. So this smallpox again was caused by a virus, the variola, whereas your cowpox was caused by vaccinia virus. And Jenner again discovered vaccination for smallpox immunity. So during that time, he observed that the dairymaids who had been infected with cowpox were immune to smallpox. So what he did was he created a vaccine from the material from the lesions in the arm of Sarah Nelmis. She was a dairymaid, and this material was injected to the arm of an 8-year-old boy. He was James Phipps. So Jenner, during that time, showed that injecting individual with material from a cowpox lesion provided protection against smallpox. So now we call this concept as cross-immunity. So cross-immunity. So this cross-immunity is a phenomenon in which exposure to one agent produces protection against another agent. And James Phipps, during that time, was exposed to smallpox. However, he did not develop the disease because he was vaccinated. And because of this, Jenner also vaccinated other children, including his own son. And this process, vaccination, it comes from the word vacca. So vacca, that's the Latin word for cow. Next one, Louis Pasteur. So Louis Pasteur is considered as the father of immunology. And from his last name, he developed pasteurization. So in this process, he described that heat could kill the bacteria. So for the temperature of pasteurization, we have 63 to 65 degrees Celsius for 30 minutes. And also, we could have 73 to 75 degrees Celsius for 15 seconds. And also, Pasteur developed the first live attenuated vaccine. So attenuated means weakened. So in other words, the microorganisms that were used in the development of these vaccines are already weakened. So they are no longer infectious whenever they are inoculated to a certain individual. But you have to take note also that this vaccine, the attenuated microorganisms in this vaccine, although they are weakened, still they are foreign. So what will happen is our immune system will still be stimulated to produce antibodies against these attenuated microorganisms. And if ever we are exposed to a certain pathogen, then there is a higher tendency that we might not get the disease because we were previously exposed to the attenuated microorganisms through vaccination. And also, in working with the bacteria that cause chicken cholera, Pasteur accidentally found out that old cultures of this bacteria would not cause disease in chickens. Also, he found out that subsequent injections of more virulent or more infectious microorganisms has no effect on the birds and other chickens that had been previously exposed to the older cultures of this bacteria. So still the concept of attenuation can be found in this instance. And also, Pasteur applied the principle of attenuation to the prevention of rabies and also anthrax. So attenuation can be done through heat, aging, or chemical means. And also, just a little story about this one. So during that time, Pasteur still has no idea on the causative agent of rabies. Talking about rabies here. But during that time, he already recognized that the central nervous system was affected whenever a person has rabies. And what happened is, he studied the spinal cords from rabid animals. So he noted that the spinal cords, if they are left to dry for a few weeks or few days, they were less infectious to the laboratory animals as compared to the fresh spinal cords. So that's the concept also in which we can find attenuation here. And there was this instance in which a boy was bitten by a severely rabid dog. So that boy was brought to the home of Pasteur. So in that time, Pasteur tried out his new procedure, this attenuation. So he gave the boy a series of 12 injections, beginning with the material from the least infectious spinal cords and progressing to the fresher and more infectious material. And miraculously, the boy survived, and now the modification of this procedure used in the past by Pasteur is considered as the standard treatment for rabies today. So other key players include Elie Metchnikov. So Elie Metchnikov is a Russian scientist who observed that when foreign objects are introduced to a transparent starfish larva, are surrounded by motile cells. So if these are the foreign objects introduced to the starfish, then they will become surrounded by motile cells, and eventually these motile cells will eat up these foreign objects, destroying them. So he called these motile cells as scavenger cells, and the process is what we call as phagocytosis. Also, we have Gerald Edelman and Rodney Porter who described the structure of antibody. Next one, Emil von Behring. So he described the action of serum antitoxin against diphtheria. For Paul Ehrlich, it has this quantitative measurements of immune reaction and chemotherapy, and also the use of salvarsan as the first synthetic antibiotic. For Jules Bordet, he described the process of complement. Rosalind Yallow for radioimmunoassay. We have Snell, Doucet, and Bernasaraf for MHC. Next one, we have Jern, we have Kohler, and we have Milstein for monoclonal antibodies, and Susumoto Tonigawa for antibody diversity, and we have Thomas and Murray for transplantation. So I'll just have some trivia here because I think this is not that well known in the field of medical technology. So who coined the term hapten? So this person, I think you already know him. So he is Carl Landsteiner. So in the field of medical technology, Landsteiner is perhaps best remembered for his discovery of the ABO blood groups and the naturally occurring ABO antibodies. And as a result of his work, the use of blood transfusions became an accepted therapeutic practice, and because of this, he was awarded a Nobel Prize in 1930. But take note that he is also the one who coined the name hapten. So this refers to simple chemical groups that could bind to antibody but which were incapable of stimulating antibody formation unless tied to a larger carrier molecules. So in short, this hapten cannot stimulate antibody production unless it is bound to a large carrier molecule, just like a protein. So again, that was coined by Landsteiner. And also, the book of Landsteiner, The Specificity of Serological Reactions, established the immunochemical basis for immunologic reactions. So those are just few of his works. Now let us have the immunity. So by definition, immunity is the sum total of the defense mechanisms of the human body to resist infectious diseases. So in other words, immunity is the condition of being resistant to infection. So it reflects how the body's immune defenses would interact to protect us from these invading pathogens that cause these infectious diseases. And for the types of immunity, we have natural and adaptive. So for natural immunity, the other term for that is innate or inborn immunity. For the adaptive immunity, it is otherwise known as acquired immunity. So for the natural immunity, it is further divided into first line and second line of defenses. The first line of defenses include the external component, which includes also the physical and biochemical barriers of the immune system. For the second line of defense, we have the internal components, which include the cellular and fluid component of the immune system. And for the third line of defense, which is under the adaptive immunity, it is further divided into cellular immunity or cell-mediated immunity and humoral immunity. So the cellular immunity or the cell-mediated immunity is mediated by T lymphocytes, whereas the humoral immunity is mediated by B lymphocytes. And both of them are further subdivided into either active or passive immunity. So let's go into detail about natural immunity. So natural immunity is otherwise known as innate or inborn immunity, or also known as non-specific immunity. Non-specific, it is because this type of immunity uses the same defense mechanism regardless of the character of the foreign antigen. And also, it provides protection by natural means without the production of antibodies. And also, it is already present at birth and it lacks memory. So take note of the two here, the production of antibody and the memory, because this differentiates your natural immunity to the adaptive immunity. Because again, in natural immunity, there is no production of antibody and there is no memory. However, in the adaptive immunity, there is production of antibody and there is already a memory. And also, this natural immunity has a uniform response. So the primary response is equal to the secondary response. So that means the mechanism doesn't alter on repeated exposure to any antigen. So again, as what I have mentioned, the natural or the innate immunity includes the first line and the second line of defense. So for the first line, we have the physical barrier and the biochemical secretions. So for the physical barrier, it is included here, the intact skin. So this intact skin includes the keratinization of the upper layer of skin and constant renewal of the skin's epithelial cells. And also, there are normal flora present in our skin that help us eliminate the invading pathogens that might cause skin infections. And also, we have the mucous membrane. So the mucus adhering to the membranes of the nose and the nasopharynx in particular traps the microorganisms. So these microorganisms that have been trapped in the mucus can then be expelled through cough reflex or sneezing. Next one, the cilia. So these hair-like structures, which are particularly found in our respiratory tract, they have this what we call as the rhythmic waving and beating motion that keep our airways clear of mucus and dirt. So that means it would help us allow to breathe easily and without irritation. How about for the biochemical secretions? So first, we have the lysozyme. So this lysozyme is found in tears and saliva. So one of the functions of the lysozyme is that it destroys the cell wall of the bacteria, particularly certain gram-positive bacteria. Also, we have the lactic acid found in our sweat, the sebum, the oil produced by the skin. So they are also biochemical secretions. So the sebum from the sebaceous glands and the lactic acid in sweat, both of them possess antimicrobial properties. Next one, we have the earwax or the cerumen. So also this one has lubricating and antibacterial property. And in addition to that, to these biochemical secretions, we also have the alkalinity and acidity of fluids in the stomach and intestinal tract, and also the acidity of the secretions of the vagina. Next one, for the second line of defense, we have cellular and humoral component. So for the cellular component, we have these cells. So the phagocytic cells, the monocytes, macrophages, and polymorphonuclear cells. So the monocytes, they are found in the blood. The macrophages, they are in the tissues. So for PMNs, in particular, we have the neutrophils. So, neutrophils. So, one of the main differences of the monocytes and the macrophages is that the monocytes contain peroxidase, whereas the macrophages don't have this peroxidase. And another one, in terms of phagocytosis, the macrophages may not be as efficient as the neutrophils because the motility of the macrophages is low as compared to the neutrophils. That's why if ever there is infection, the first type of cell that would go to the site of infection are the neutrophils. Next one, we have eosinophils, which is commonly increased during parasitic or allergic reactions. And also we have basophils and mast cells for allergic reactions. We also have antigen-presenting cells and natural killer cells. Next one, the humoral component. So the major component is the complement, but take note of this one. Complement of the alternative pathway, because the alternative pathway of complement does not involve antibody production. But the classical pathway of the complement is the one that involves the antibody. That's why this alternative pathway is specified here, because we are talking about natural immunity that does not include antibody production. Next one, we also have the cytokines like the interferon and interleukin. So interferon, they are like natural antibiotics. So they are family of proteins produced by many cells in response to viral infection. So they are called as interferon because they block or they interfere with the replication of the virus in other cells. Next one, we also have the lysozymes again, and also the properdine. So the properdine has bactericidal and virucidal property, and also this properdine can activate complement. Next one, beta-lysine. So this beta-lysine is a substance that is released by platelets during coagulation, and also this is to help us eliminate gram-positive bacteria, of course, except streptococci. So eliminate gram-positive bacteria except streptococci. So that is for beta-lysine. Next one, we have acquired or adaptive immunity. So this one is a specific type of immunity because again, this already includes antibody production. So unlike your natural immunity, this one is not present at birth. So we are born with the ability to produce an acquired immunity, but the response is not immediately available. So this is a diverse type of response because the secondary response is greater than the primary response, because again, our immune system would tend to develop antibodies against specific antigens. And also, it has memory, unlike your natural immunity, because it can recall previous antigens. Now for the third line of defense. So this is again included in the acquired or adaptive immunity. So it has the cellular and humoral component. So let us have first the humoral component. So when we say humoral immunity, this is a type of immunity that results from the formation of antibodies. So if a microorganism already overwhelms the body's natural immunity or natural resistance, there is this third line of defense that will come into picture. So this type of acquired or adaptive immunity is a mechanism that would allow us to recognize, to remember, and to respond to a specific antigen. So again, to recognize, to remember, that's why there is memory here, and to respond to specific antigen. So that's the main functions of the acquired or adaptive immunity. So this adaptive immunity can then result in the elimination of microorganisms and of course the recovery from disease, and the host often acquires a specific immunologic, again, memory. So there is now memory in the acquired or adaptive immunity. So this condition of memory, or oftentimes we call this as recall, so this is an acquired resistance that allows the host to respond more effectively if reinfection with the same microorganism or same antigen occurs. So also in the humoral component, we can have the cytokines and complement. Take note of this one, the classical pathway of complement, because this one involves the function and the action of antibodies, whereas the alternative complement pathway does not involve the antibody formation. Next one, for the cellular component, so we have specialized lymphocytes. We have the T lymphocytes, B lymphocytes, or the activated B lymphocytes, which is the plasma cell. So these are the ones that produce the antibodies, the plasma cells. Also, we have the antigen-presenting cells such as the monocytes and the macrophages. So in the cellular component, the major component of the acquired immunity is the lymphocyte. So this is the major cellular component of acquired immunity. And the major humoral component is the antibody. So acquired immunity can be conferred in two ways, the active and the passive immunity. So take note of this basic principle. When we say active immunity, there is the introduction of antigen. Whereas when we say passive immunity, there is the introduction of an antibody. So in short, in active immunity, because we are given antigen, it is our body or it is our immune system that would produce antibody. However, in the passive immunity, our body is not the one producing antibodies because again, the antibodies are given to us. So for the active immunity, again, there is an introduction of antigen, and the antibody production is done by the body. So since our antibody is produced by the body and our immune system is stimulated because of the antigen that was introduced to us, the type of immunity conferred by this active immunity is long-term. But again, since we are giving time to our body to develop the antibodies, the immune response is slow. So it can be a natural active immunity or artificially active immunity. Natural means by having an actual infection, by getting sick or acquiring diseases naturally. But when we say artificial immunity, that could be through vaccination. And I hope you still remember who discovered this one. So active immunity was first demonstrated by Edward Jenner. So the artificial active immunity includes the inoculation of live organisms like in smallpox. We also have attenuated or weakened organisms in Bacillus calmette-guerin, in Mycobacterium tuberculosis, and also the Mycobacterium bovis, and also inoculation of dead organisms like in cholera and typhoid fever. Next one, we have the toxoid, so like in Clostridium tetani, and the modified virus like the poliovirus. So these are all examples of artificial active immunity. So in passive immunity, there is again introduction of antibodies, and that the antibody production is not done by the body. And because of this, the immune response for passive immunity is immediate. So just like, for example, when you are bitten by a dog, so you won't wait for your body to produce antibody for weeks or for days after bitten by a dog. But what will happen is you will be given an immediate source of antibody to counteract the effect of the rabies virus. But because you did not give your body a time to produce antibodies, then the immunity is just short-term, unlike your active immunity. So, like your active immunity, this could also be a natural passive immunity or artificial passive immunity. So, for natural immunity, we have transfer of antibody in vivo. So, just like in pregnancy, because some classes of IgG could cross the placenta. So, the babies will be protected by the antibodies coming from the mother. Also, in breastfeeding, we have here the colostrum, which is rich in immunoglobulin A. So, it's a milky fluid that is released by mothers that have recently given birth before breast milk production begins. So, it's an important source of nutrients that also promotes growth and fight diseases in infants. So, that's for colostrum. Next one, artificial passive. So, again, there is an administration of antibody. So just like in my example, if you are bitten by a dog, then immediately you will be given anti-rabies. So this is containing antibodies. Also immunoglobulins against Hep B and also ROGAM. So this is an antibody given to mothers who are Rh negative. So that's an example of artificially passive immunity. So this one just shows the differences between the active and passive immunity. So again, both of them could be acquired through natural or artificial means. And again, in active immunity, the antibody production is done by the body, whereas it's not done by the body in passive immunity. And also, the immune response in active immunity is long-term, whereas in passive, it's short-term. But you also have to consider if the host or the person is immunocompetent or not, and also the half-life and the lifespan of the antibodies and the cells involved here. Now let us elaborate the types of adaptive immunity. So again, we have the humoral and cell-mediated immunity. So the humoral response is carried out by antibodies produced by plasma cells. And again, these plasma cells are derived from activated B cells that are produced in the bone marrow. So with the assistance of T helper cells, this one, the B cells now will differentiate into plasma cells, which can produce antibodies against a specific antigen. So what will happen is that the antibodies produced by the plasma cells will now bind to the antigen. And what will happen to this antigen after binding is that it will be neutralized or it will be lysed through dissolution or destruction of cells by a lysine or even through phagocytosis. So that is what will happen to the antigen. So remember, the aim of pathogens or antigens is to enter the cell and to destroy it. So that's where the antibodies come into the picture by interrupting the receptor linkage between the pathogen and the cell. So now the result will be the breakdown of interaction between the pathogen or the antigen and the cells. And also, you have to take note that the humoral immune system deals with antigens from pathogens that are freely circulating or outside the infected cells. So that means this type of immunity deals with extracellular pathogens. On the other hand, the cell-mediated immunity is a principal defense mechanism against intracellular microbes. So that is also one of the differences of the cell-mediated and humoral immunity. Now, how about for cellular immunity? So this one is mediated by T lymphocytes that arise from the bone marrow and mature within the thymus gland. So let me just emphasize that one. So again, the production site of these T lymphocytes is in the bone marrow. But for maturation, it is in the thymus gland. Okay, so after maturation in the thymus gland, what will happen is these T lymphocytes now will express a unique antigen binding molecule. So that is what we call as the T cell receptors. So these are cell membrane receptors that bind to non-self substances presented on the surface of antigen-presenting cells bound with MHC molecule. So for example, you have here a mature T cell. So again, it has membrane receptors here. So that receptors will be recognized by the antigen presenting cell. So this one, antigen presenting cell, which is attached to an MHC molecule. So the antigen that was processed by the MHC will be expressed by the antigen presenting cell. And this antigen presenting cell now will present the antigen to the receptors found in the T lymphocytes. So that's the function of the receptors and of course the antigen presenting cells. Because you have to take note here that unlike antibodies, the T cells can recognize antigens only when it is presented along with the MHC on the membrane of the antigen presenting cells. And so after binding, T cell activation will take place and of course there will be the destruction of this antigen. So let's take a look at how the cellular immunity works. So remember, cellular immunity is the major defense against intracellular pathogens. So in this case, the T cells are created to destroy infected cells of our own body. So for example, if a person is infected with a pathogen, example with a virus, there are these unique antigens on the outer surface of the pathogen. So this one, these are the unique antigens found in the surface of the virus, for example. So these are proteins outside the cells and viruses that would also help us to identify and to mark the cells that belong to us and those that are foreign to us. So what will happen next is that the pathogen will infect or enter the body cells. Remember, cellular immunity is the major defense against intracellular pathogen. And after entering the cell now, the antigens on the pathogens are displayed on the outside of the infected cells of the body. So just like this one. So the antigens are displayed outside of the cells. And what will happen next is that the other pathogens here, so they will be destroyed or will be eaten by a phagocyte. And the phagocyte now will destroy these pathogens and this phagocyte will present or display the antigens of the destroyed pathogen on its surface. So after displaying that one, so this phagocyte now will become an antigen presenting cell. So that is why they are called as such, because they present the antigen on its surface. So after presenting the antigen, what will happen is that the inactivated T cells will bind to the antigen presenting cells. And after that, the antigen presenting cells will release proteins that will be picked up by the inactivated T cells. So they will release proteins, and what will happen is that this T cell will divide into a memory T cell and active or plasma T cells. So the memory T cell will fight pathogen in the future. However, the active plasma T cells will bind to the infected body cells. And after binding to the infected body cells, what will happen is that these active plasma T cells will release proteins and also enzymes to destroy the infected cells. So we call them as cytotoxic T cells. And after that, of course, the pathogen will be destroyed, and of course, the healthy neighboring cells will multiply through mitosis. So that is how your cellular immunity works. So now let us take a look on how the humoral immunity processes the antigen. So again, in this type of immunity, B cells are created to produce antibodies. So for example, if a person is exposed to a pathogen, the membrane-bound antibodies of a B cells will bind to pathogens. So this is a B cell, and it has this membrane-bound antibodies, and that antibodies will bind to the pathogen. Next one, the pathogen will be engulfed by the B cells, and it will be broken down. And after that, after the antigen has been broken down by this B cell, the antigen of the pathogen will be presented on the surface of the B cell. So this one now, this B cell will act as an antigen presenting cell or an antigen presenting B cells. So after presenting the pathogen, what will happen next is that the helper T cells will bind to the antigen-presenting B cells. And these T helper cells will release proteins to activate the B cells. And once the B cells are activated, they will divide into memory B cells or active B cells. So the memory B cells now are used to fight pathogens in the future, whereas the active B cells are the ones producing antibodies to attack the pathogens. So those are the functions of those memory B cells and active B cells. So once the antibodies are produced and released in the plasma, there are three things that might happen. So number one is that the antibodies will attach to the antigens of the pathogens. So that will prevent or will interfere the pathogen from infecting another cells. Or they may clump or they may attach to multiple pathogens together. So they will clump or attach the multiple pathogens together, and what will happen next is this pathogen will be tagged as foreign. So it will be easily recognized by our immune system. Or lastly, the phagocytes will swallow and destroy the pathogen, so the current infection is defeated by the immune system. So that is how our humoral immunity works. So that's it. I hope everything has been clarified to you. Thank you.